A very good evening to all. Today we are going to discuss cell and some important events regarding the cell cycle. So let's start with the cell. Before we proceed with the lecture, let's discuss a question. Given below are two statements. Statement 1, the membrane-bound organelles of the endomembrane system coordinate cellular function. Statement 2, mitochondria, chloroplast are not considered a part of the endomembrane system. In the light of the above statement, choose the most appropriate answer from the option given below. Option 1 says that statement 1 is incorrect and statement 2 is correct. Option 2 says both statement 1 and 2 are correct. Option 3 says both statement 1 and 2 are incorrect. And option 4 says that statement 1 is correct, but statement 2 is incorrect. So quickly answer what you think, what will be the correct option for this question. The statement number 1 says that the membrane-bound organelles of the endomembrane system coordinate cellular function. Membrane-bound organelles, that means they are talking about ER, Golgi, lysosome, and vacuoles. These are components, the ER, Golgi, lysosome, and vacuole. They are the part of the endomembrane system. Why? Because they show coordinated function. For example, if this is a nucleus, the outer membrane of the nucleus is continuous with the endoplasmic reticulum. So the vesicle will bud out from the ER, and this vesicle is containing the protein. Now this vesicle will fuse with the cis phase of the Golgi. And this is the trans phase. Now the vesicle, which is containing the protein, will fuse with the cis phase of the Golgi, and in the Golgi, the modification of the protein takes place. And after modification, the vesicle containing the modified protein will be bud out from the trans phase. Now, if this vesicle contains the hydrolytic enzyme, then this vesicle is known as a lysosome. So you can see that the ER, Golgi, lysosome, they all are showing the coordinated function. So the membrane-bound organelles of the endomembrane system, they show a coordinated function. Whereas the mitochondria and the chloroplast, they are not considered as a part of endomembrane system. Because the mitochondria and the chloroplast, they does not show the coordinated function. So both statement 1 and statement 2 are correct. So the correct option is option number 1, which says that statement, sorry. So both statement 1 and statement 2 are correct. So correct option is option number 2, which says both statement 1 and statement 2 are correct. Now let's discuss the cell theory. As we all know that the cell is the basic unit of life. And the cell theory was proposed by Schleden and Schwann. Schleden was the botanist and Schwann was the zoologist. So Schleden, he worked on the plants and he observed that the plants, they composed of different type of cells, and these cells, they form the tissue in the plant. Schwann worked on plants and animals. Schwann, while working on plant, he find out that the plants, they have a unique character, and that character is a cell wall. So the plants, they have a cell wall. In animal, while working on the animal, Schwann proposed that the animals, they have a plasma membrane. So the Schwann proposed a hypothesis that the animals and plants, they are composed of cell and the product of cell. So Schwann and Schleden together, they formulated the cell theory. But this theory could not explain that how the cells are formed, that how the cells arise. So there's a third scientist named as Rudolf Virchow. In 1855, Rudolf Virchow proposed a statement which says omnicellular e cellular, which means that all the cells are arise from the pre-existing cell. So today, the modern cell theory is composed of 2 points, that all the living organisms are composed of cell and the product of cell. And second, that all cells arise from the pre-existing cell. Now let's discuss the prokaryotic cell. We know that there's a two type of cells, prokaryotic cells and the eukaryotic cells. Prokaryotic cells, they are generally simpler and they multiply more rapidly. So prokaryotic cells, they have a cell envelope, and cell envelope, it consists of three layers. Number one is a glycocalyx. Glycocalyx could be a capsule or a slime layer. If it is capsule is thick and tough, so it cannot be easily washed off, whereas slime layer is loose sheath, so it can be easily washed off. Inner to the glycocalyx, a cell wall are present, and in case of bacteria and prokaryotes, the cell wall are made up of peptidoglycan. Inner to the cell wall is the cell membrane. Cell membrane is present, and the cell membrane in prokaryote as well as in eukaryote, they are similar. They are selectively permeable. They are phospholipid bilayer in nature. And the genetic material in case of prokaryote is circular, double-stranded, closed, and naked. Naked, why? Because they lack histone protein. And they do not have a well-defined nucleus because they lack nuclear membrane. Prokaryote, they have plasmid, which is an extra chromosomal DNA and which impart the unique phenotypic character to the bacteria, that is antibiotic resistance. Bacteria, they do not have a membrane-bound organism. The membrane-bound organisms like Golgi, lysosomes, vacuoles, mitochondria, chloroplast, they all are absent in case of prokaryote because it does not have the membrane-bound organelles. But the infoldings of the plasma membrane in case of prokaryotes, they lead to a formation of a structure which is known as a mesosome. So mesosome perform a very important function, that is the cell wall formation, DNA replication, also in the respiration because it contains the respiratory enzymes. Prokaryotes, they have a chromatophore. Chromatophores are the structure that contains the pigment like cyanobacteria. Cyanobacteria is a blue-green algae. It is photosynthetic in nature. It performs photosynthesis. So cyanobacteria, they have chlorophyll A, and this chlorophyll A pigment is present in a structure which is known as chromatophore. Prokaryotes, they have a more organ which is known as flagella, which provide the motility to the cell. And the flagella, it composed of three parts. There's a filament, hook, and the basal body. The basal body is embedded in the cell wall or the cell membrane. And the bacteria or the prokaryotes, they have flagella. The flagella, they are larger in size. You can see they're longer. And the fimbriae and pili are present. Pili, they are made up of special protein which are known as pillin. The pili, they are generally larger in size as compared to the flagella, and the fimbriae are present. Fimbriae are the bristle-like structures which are present on the surface of the prokaryotes, and the function of the fimbriae is the attachment to the substratum. They help the prokaryotes or the bacteria to attach to the surface of the substratum. Prokaryote, they also have inclusion bodies. Inclusion bodies, they are the special structures which has the reserved material and they lie freely in the cytoplasm. These inclusion bodies like phosphate granules, cyanophycine granules, glycogen granules, or gas vacuoles. Gas vacuoles are found in purple and green sulfur bacteria or in blue-green algae, and they provide buoyancy to the bacteria. In prokaryotes, the ribosomes are 70S. Now let's discuss the structure of the cell membrane. As I told you, in prokaryotes as well as in eukaryote, the cell membrane is selectively permeable in nature. It is made up of the phospholipid bilayer. They have the polar head and the non-polar tail. The non-polar tail is projected inward and the polar head are projected outward so that the polar head can protect the non-polar tail from the aqueous environment. Now, the chemical studies on the membrane of human RBC was done, and it showed that the membrane are composed of lipids and proteins. So there are two types of proteins are present, the peripheral protein and integral protein. Peripheral protein which are located at the periphery, and integral proteins which are partially or totally buried in the membrane. And the membrane, they also have cholesterol. Main function of the cholesterol is to maintain the fluidity of the membrane. Various model has been proposed regarding the cell membrane, like unit membrane model, sandwich model, but the most accepted model is the fluid mosaic model, which was proposed by Singer and Nicholson. According to this model, the membrane has the quasi-fluid nature. The quasi-fluid nature, quasi means semi, so they are semi-fluid in nature. And the lateral movements of protein occur within the lipid bilayer, which shows the fluidity of the membrane. And the membrane, it performs different functions like cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division, etc. The most important function that is performed by the cell membrane is the transport of the molecule across it, like passive and active transport. You can see this in this figure. This is a one molecule of phospholipid showing the phosphate head, which is a polar head, and the non-polar or the hydrophobic tail. Now the cell wall. Cell wall, they are the dead structure and they are permeable. Like the molecules, they can easily pass through the cell wall. Cell wall, they provide shape and protect the cell from mechanical damage. They also help in the cell-to-cell interaction and provide a barrier to the undesirable macromolecule. Cell wall in case of algae, they are made composed of cellulose, galactone, mannans, minerals, and like calcium carbonate. And in higher plants, the cell wall is composed of cellulose, hemicellulose, pectin, and proteins. In young plants, they have a primary cell wall. So as the plant matures, the primary cell wall starts degenerating and the secondary wall is laid down. Always remember that secondary wall is laid down inner to the primary wall. So you can see this. There's a primary wall, and inner to the primary wall, the secondary wall is laid down. And inner to the secondary wall, there's a presence of plasma membrane. And the two cells, the two neighboring cells or the adjacent cells, they are joined together through a cementing agent, which is known as a middle lamella. And this middle lamella is made up of calcium pactate. And the cytoplasm of the two cells, if this is a cell, the cytoplasm of the two cells are connected through the cytoplasmic bridge, and these cytoplasmic bridge are known as plasmodesmata. Next one is endoplasmic reticulum. We know that the prokaryotes, they lack membrane-bound organelles, but the eukaryote have the membrane-bound organelles. And these membrane-bound organelles are endoplasmic reticulum, the Golgi bodies, lysosome, and vacuole. These are the four membrane-bound organelles which forms the endomembrane system. They form the endomembrane system because they show the coordinated function. So endoplasmic reticulum was first observed by the Keith Porter. And endoplasmic reticulum, it has two distinct compartments. There's a luminal, which is the inner side, and the extraluminal, which is the outer side. And the ER, which is associated with ribosome, is known as rough ER. And the function of the rough ER is involved in protein synthesis. And the ER without ribosome is known as a smooth ER or the smooth endoplasmic reticulum. And the function of the smooth endoplasmic reticulum is the lipid synthesis or steroid hormone synthesis. SER is also involved in drugs detoxification because it has a cytochrome P450. Remember this, it's very important. And also remember that the outer membrane of the nucleus is continuous with the ER. Next one is Golgi apparatus. Golgi was discovered by Camillo Golgi in 1898. They consist of many flat and disc sac or the cisterna of 0.5 micrometer to 1 micrometer diameter. Golgi, they have two phases. They have a cis phase, convex phase, or the forming phase. And the second phase is the trans phase, maturing phase, or the concave phase. So the cis phase is the one which receive the vesicle containing the lipid or the protein from the ER. They get fused or they get received by the cis phase. Now, these middle, they are known as the medial cisterna, where the modification of the protein or lipid takes place. And now the modified protein and the lipid, they bud out from the trans phase. Okay. Now, this is the vesicle which contains the modified lipid and protein. The modified lipid and protein are known as glycoproteins or the glycolipids. So main function of the Golgi is is in the packaging of the material, and second one is the modification of the protein and the lipids. Next one is lysosome. Lysosome, they are membrane-bound vesicles, and they're formed by the packaging, by the process of packaging in the Golgi apparatus. As I told you earlier, that when the vesicles burst out from the transphase of the Golgi, if the vesicle contain the hydrolytic enzyme, then that vesicle will function as a lysosome. And the enzymes which are present in a lysosome, they are hydrolytic enzymes. So they are active at acidic pH. Vacuole, they have a membrane which is known as a tonoplast. And in case of the plants, the vacuole, they occupy 90% of the portion of plant cell. And these vacuole, they contain water, sap, excretory product, and other material which are not useful for cell. In case of amoeba, the contractile vacuole are present, and the function of contractile vacuole is in excretion and osmoregulation. Osmoregulation means that they maintain the osmolarity of the cell. Second, the protists, they have the food vacuole, and the function of the food vacuole in protists, the food vacuole are formed by the engulfing of the food particles. Next, please remember one more important point, that endoplasmic reticulum, Golgi apparatus, the lysosome and vacuole, they all are single membrane bound. Okay, they all are single membrane bound, and they're part of endomembrane system. Mitochondria, it is known as the powerhouse of the cell. Why? Because it synthesizes the ATP. Its main function is the ATP synthesis. So mitochondria is a double membrane bound organelle. It has double membrane. There's the outer membrane and the inner membrane. The inner membrane, it shows enfolding, and the enfoldings are known as cristae. And the inner membrane of the mitochondria have the enzymes that play a very important role in the ETS, that is the electron transport chain. The fluid which is present inner to the inner membrane, that fluid is known as a matrix. And the mitochondria, they have a prokaryotic origin because they have 70S ribosome, they divide by fission. They have a genetic material similar to the prokaryote, that is double-stranded and circular DNA. So we say that the mitochondria as well as the chloroplast, they both have the prokaryotic origin. And mitochondria is the site for ATP synthesis. It's the site where the aerobic cellular respiration takes place, the site where the Krebs cycle occurs, the site where the ETS, the electron transport chain, occur. So mitochondria is the site for ATP synthesis. That is why we call mitochondria as the powerhouse of the cell. Next one is plastid. So plastids, they are found in plant cell and in lignioids. There are three types of plastid. There are chromoplast, chloroplast, or leucoplast. Chromoplast, they contain carotenoids and xanthophyll. Chloroplast, they contain chlorophyll and carotenoids. Leucoplast, they are colorless plastids. And leucoplast, they're further divided into amyloplast. Amyloplast that store carbohydrate, aleuroplast that store oils and fat, and eleuroplast that store proteins. Today, we are going to discuss chloroplast in detail. So main function of the chloroplast is to perform photosynthesis, right? And the number of the chloroplast, it vary, like 1 in Chlamydomonas, which is a green algae, to 20 to 40 in a mesophyll cell. And chloroplast is a double membrane structure. It consists of outer membrane and the inner membrane. The inner compartment is filled with the fluid which is known as stroma in case of chloroplast. The chloroplast, they have a sac-like structure which are known as thylakoid. And these sac-like structure, they stack over one another and they are known as granum. And two different granums, they are connected to each other by the tubule which is known as a stroma lamellae. As chloroplasts, they also have a prokaryotic origin. So they also have a ribosomes, which are 70S, and the genetic material is similar to prokaryote, like small double-stranded circular DNA. Ribosome was first observed by George Palade in 1953. The main function of ribosome is protein synthesis. They consist of two subunits, there's large subunit and small subunit. In case of prokaryotes, the ribosome are 70S. In case of eukaryotes, the ribosome are 80S. And the S stands for Sedgwick unit and also stand for the sedimentation coefficient. And it is an indirect measure of density and size, because on adding 50 and 30 we get 80, but we are getting 70S because it's not additive. It is the indirect measure of density and size. That is why we are getting 70S. And please remember, in case of prokaryotes, the cytoskeletons are absent. In prokaryotes, cytoskeletons are absent. In eukaryotes, we see three types of cytoskeleton. They are microtubules, microfilaments, and intermediate filaments. And the function of these cytoskeleton is they provide mechanical support, they provide motility and maintenance of the shape of the cell. Next, we are going to discuss cilia and flagella. The flagella of prokaryotes are differ from the eukaryotes because the flagella of prokaryotes, they are made up of a protein which is known as flagellin proteins. And they have structurally, they are different. Whereas in eukaryote, the flagella, they have 9+2 arrangements. Remember this, the cilia and flagella of eukaryote, they have a 9+2 arrangement. So this is a structure of exoneme. You can see there's a 9 peripheral doublets. There are 9 peripheral microtubule doublets, and the 2 singlet are present in the center. And this green portion, these are the central sheath. So these 9 peripheral microtubule doublets, they are connected to the central sheath by the radial spoke. These green color, these are radial spoke. And the peripheral doublets are interconnected by the linkers. And both cilium and flagellum, they arise from a centriole-like structure called as basal body. Next is the centrosome and the centriole. So the centrosome, they are composed of 2 perpendicular centrioles. Okay, these are the centrioles. They are perpendicular to each other. And the centriole plus the amorphous pericentriolar material, they together form the centrosome. And centrosome play a very important function in cell division. So the centriole, they show 9+0 arrangement. 9 are the peripheral microtubule triplets. Remember, in case of cilia and flagella, there were 9 peripheral doublet, but in case of centriole, there are 9 peripheral microtubule triplets. And in the center, there's no singlet is present. In case of cilia and flagella of eukaryote, they're showing 9+2 arrangement. There were 2 singlet are present in the center. But in case of centriole, there's no singlet. So there's only, so they show arrangement of 9+0, where there's the 9 peripheral microtubule triplets. And these peripheral microtubule triplets, they are connected to the central hub by the radial spoke. The centriole, they form the basal body of the cilia and flagella and the spindle fiber that give rise to the spindle apparatus during the cell division in animal cell. One more important point, that the plant cell, they lack centriole. Plant cell does not have centrioles. Remember this. Moving next is the nucleus. So nucleus is also a double membrane structure. It has an outer membrane and the inner membrane. And the space between the two membrane is known as a perinuclear space, which is 10 to 15 nanometer in diameter. And the nucleus was first observed or discovered by Robert Brown, chromatin by Fleming, and the interphase nucleus contains chromatin. So when the cell is in the interphase, they have chromatin. But when the cell reaches to the cell division phase, that is the M phase, now this chromatin start condensing or compacting to form a structure that is known as a chromosome. Now the chromatin has condensed and compacted to form the chromosome during the division phase, during the nuclear division phase or the M phase. And during the interphase, the nucleus that contain chromatin. The outer and the inner membrane of the nucleus, they are interrupted by the nuclear pores. So their nuclear pores are present. Here, their nuclear pores are present. These are the nuclear pores through which the molecule can move out of the nucleus. And the erythrocytes of many mammals and sieve tube cells of vascular plants, they lack nucleus on maturity. Remember this, that the mature RBC and the mature sieve tube cells of the vascular plants, they lack nucleus. And inner to the fluid in the nucleus is known as a nucleoplasm. So nucleoplasm is consist of nucleolus and chromatin. Nucleolus is a non-membrane bound. It does not have a membrane. So the content of nucleolus is continuous with the nucleoplasm. And the nucleolus is the site for rRNA synthesis. Remember this, very important, that nucleolus is the site for rRNA synthesis. And we know that the ribosomes, they are made up of RNA and protein. So nucleolus is also known as a ribosome factory. So chromatin, they contain DNA, some basic proteins called histones, some non-histone proteins, and also RNA. So their primary construction is present, which is known on the chromosome. This is known as centromere. The main function of the centromere is to hold the sister chromatid together. And on the position of the centromere, the chromosomes are of 4 types. They can be metacentric if the centromere is present in the center and dividing the chromosome arms into equal. That means the length of the P arm and the Q arms, they are equal because the centromere is present in the center. In submetacentric, when the centromere is slightly away from the center, then the chromosomes are known as submetacentric. When the centromere are present slightly towards the terminal end, then it is known as acrocentric. And when the centromere are present at the terminal position, then the chromosomes are known as telocentric. And the secondary constructions are also present, which are known as satellite. Few chromosomes, they have a non-staining secondary construction at a constant location. So these satellite, they have repeated DNA. And these repeated DNA, they are non-coding DNA. That means it does not code for any protein. They show high degree of polymorphism. Please remember these points, these are very important. And on the centromere, the disc-like structure are present on both the side of the centromere, and these are known as kinetochore. So during the metaphase, the spindle fibers from the opposite end, they attach to the kinetochore of the chromosome. Next are the microbodies. They are the membrane-bound minute vesicles. They contain various enzymes. They are found in both plants and animals. Example, peroxisomes, spherosomes. Now, let's discuss some of the questions. Question says that which of the following pair of organelles does not contain DNA? Which of the following pair of organelles does not contain DNA? I told you that mitochondria and chloroplast, they have a double-stranded and circular DNA. Right? Because mitochondria and chloroplast, they have a prokaryotic origin. And they're also known as a semi-autonomous organelles. Option 1 says mitochondria and lysosome. Mitochondria have DNA, lysosome does not. Option 2 says chloroplast and vacuole. Vacuole does not have DNA. Option 3 says lysosome and vacuole. Option 4 says nuclear envelope and mitochondria. Mitochondria has DNA and nuclear envelope, that means they are referring to nucleus. Yes, nucleus also have DNA. But lysosomes and vacuole, they does not have DNA. And question is asking which of the following pair of organelles does not contain DNA? Does not, okay? So lysosome and vacuole, they does not contain DNA. Moving next is which of the following cell organelle is responsible for extracting the energy from carbohydrate to form ATP? Option 1 is ribosome, option 2 is chloroplast, option 3 is mitochondria, option 4 is lysosome. Which of the following cell organelle is responsible for extracting the energy from carbohydrate to form ATP? Main function of ribosome is in protein synthesis. Chloroplast, they perform photosynthesis in plants. Mitochondria, ATP synthesis. And the question is asking which of the following cell organelle is responsible for extracting the energy from carbohydrate to form ATP? So cellular respiration involves a first step in cellular respiration is glycolysis, where glucose is converted to pyruvate. Now this pyruvate will enter into mitochondria, where this pyruvate is converted to acetyl-CoA. Now this acetyl-CoA will enter into the Krebs cycle, which occur in the matrix of mitochondria. This Krebs cycle will produce NADH and FADH2. Now these NADH and FADH2 will enter into the electron transport chain, which occur in the inner membrane of the mitochondria, and it leads to the formation of ATP. That is why the mitochondria is known as the powerhouse of the cell, because this aerobic respiration takes place, and it is the site where the ATP synthesis occurs. So the organelle which is responsible for extracting the energy from carbohydrate to form ATP is mitochondrion. Lysosome contain the digestive enzyme. They contain the hydrolytic enzymes. Next question says the non-membranous nucleoplasmic structures in nucleus are the site for active synthesis of non-membranous nucleoplasmic structure in nucleus are the site for active synthesis of. Option 1 is protein synthesis, option 2 is mRNA, option 3 is rRNA, and option 4 is tRNA. So the non-membranous nucleoplasmic structure, that means they are talking about nucleolus. Nucleolus is non-membrane, because of which the content of the nucleolus is continuous with the nucleoplasm. And nucleolus is the site for rRNA synthesis. And it is also known as a ribosome factory. So correct option is option number 3, which is rRNA. Next, we are going to discuss some of the important events regarding the cell cycle. First, we are going to discuss interphase. This is also known as a preparatory phase, where the cell prepare for the cell division. Interphases consist of G1 phase, where the cell growth occurs. G1 is followed by S phase, where the DNA duplication occurs, and the centrosome duplication also occurs. S phase is followed by G2 phase. In G2 phase, the protein are synthesized, which are required for the M phase. And there's also one important phase, which is Z0 phase, which is also known as a resting phase. The cell which does not divide, they exit the G1 phase and they enter into the resting phase, which is also known as a quiescent phase. This is the inactive stage. After the G2 phase, the cell enter into the M phase. And the M phase consists of two parts. There's karyokinesis and cytokinesis. Karyokinesis is a nuclear division, and cytokinesis is the division of the cytoplasm. If the nuclear division keep on happening and the cytoplasm does not divide, then it leads to a condition which is known as a multinucleate condition, and that condition is known as syncytium. So karyokinesis, they further have 4 stages. There's prophase, there's metaphase, there's anaphase, and the last one is telophase. First one is prophase. So during the prophase, the nuclear membrane disappear, nucleolus disappear, cell organelles, they start disappear, and the centrosomes which are duplicated during the S phase, they start moving towards the opposite pole. As you can see in this figure, the centrosome, they start moving towards the opposite pole, and the nuclear membrane disintegrated. And during the prophase, the chromatin, they start condensing or compacting to form the chromosome. During the metaphase, the compaction of the chromatin or the chromosomes, they are clearly visible. Or the condensation of the chromosome is completed at the late prophase or the early metaphase. And the morphology of the chromosome, they are clearly visible or study in case of in metaphase. And in metaphase, the chromosome, they get aligned at the equatorial plane. As you can see in this image, that all the chromosomes, they'll align at the equatorial plate, and the spindle fiber from the opposite pole, they get attached to the kinetochore of the chromosome. So spindle fiber from this pole will attach to the kinetochore. Spindle fiber from this pole also attached to the kinetochore. And this is the equatorial plate. During the anaphase, now the centromere divides and the sister chromatid, they get separated and they start moving towards the opposite pole. And during the telophase, the nuclear membrane reappear. the nucleolus reappear, the cell organelles, they reappear during the telophase, and the chromosome, they start decondensing to form chromatin. Telophase is followed by cytokinesis, where the cytoplasm divide, and from the one parental cell, two daughter cells are formed in case of mitosis. So the significance of mitosis are, number one, that the daughter cells are identical, genetically identical to the parent cell. That means their clones are formed. Clones, when the daughter cells or the offsprings, they are morphologically and genetically similar to their parent cell. So in mitosis, the daughter cells are genetically similar to the parent cell. Second, that the mitosis, they maintain a nuclear cytoplasmic ratio, that when the cells divide, the nuclear cytoplasmic ratio of the cell get disturbed. So to restore the nuclear cytoplasmic ratio, cell again divide. So in mitosis, they play a very important role in maintaining the nuclear cytoplasmic ratio. Next, we are going to discuss meiosis. In meiosis, two nuclear division occur. Two cell division, but single DNA replication. Remember, that's very important. And there's meiosis 1 and meiosis 2. And the gap between the two meiosis is known as interkinesis. And this gap is very short. So prophase 1 of meiosis 1 is very long and complex, because the prophase 1 of meiosis 1, it consists of 5 stages. Number 1 is leptotene. During this stage, the chromosomes are gradually visible. Second one is zygotene. In this stage, the synapses occur. The pairing between the homologous chromosome occur, and they form a synaptonyme, and the complex is formed between the homologous paired chromosome. That complex is known as a synaptonymal complex. Third is the Pachytene stage. During the Pachytene stage, the bivalents and the tetrad, which are formed in the zygotene stage, they are clearly visible in the Pachytene stage. And in the Pachytene stage, the recombination takes place, that the crossing over occur between the non-sister chromatid of the homologous chromosome, and the formation of recombination nodule occur. This reaction, or this crossing over, is catalyzed by an enzyme which is known as a recombinase. Fourth one is diplotene. During diplotene stage, the synaptonymal the terminalization or the dissolution of the synaptonymal complex takes place, but the chromosomes, they remain attached at the recombination nodule, or they form a X-shaped like structure, which is known as a chiasmata. After diplotene, there is dikinesis. So during dikinesis, terminalization of chiasmata occur, nucleolus disappear, disintegration of nuclear membrane occurs, and the centrosome, they start moving towards the opposite pole, and the condensation of chromatin start. Prophase 1 is followed by the metaphase 1. In the metaphase 1, the homologous paired chromosome, they get aligned at the equatorial plate, and the spindle fiber from the opposite pole, they get attached to the kinetochore of the homologous paired chromosome. During the anaphase 1, the paired homologous chromosome, they get separated. Now the homologous chromosome, they start moving towards the opposite pole. In telophase 1, the nuclear membrane reappear, nucleolus reappear, the cell organelles, they reappear, and the chromosome, they start decondensing to form chromatin. Next one is meiosis 2. Meiosis 2 is similar to mitosis, which is an equational division, but meiosis 1 is a reductional division. So in the prophase 2, similarly, similar as mitosis, the nuclear membrane, they disappear, nucleolus again disappear, and the chromosome, they start condensing. In metaphase 2, the chromosome, they get aligned at the equatorial plate, and the spindle from the opposite pole, they attach to the kinetochore of the sister chromatid of the chromosome. During the anaphase 2, now the centromere split and the sister chromatid, they start moving towards the opposite pole. Remember, in anaphase 1, the homologous paired chromosome gets separated, and the homologous chromosome, they start moving towards the opposite pole. In anaphase 2, the sister chromatid gets separated and they start moving towards the opposite pole. In telophase 2 and cytokinesis, first, let's discuss telophase 2. In telophase 2, the nuclear membrane, they reappear, nucleolus reappear, and the chromosome, they start decondensing to form chromatin. Now telophase 2 is followed by cytokinesis. So the end product of meiosis, we get 4 haploid daughter cell. So the major difference between the mitosis and meiosis are that the end product of mitosis is the 2 daughter cell. In meiosis, the end product are the 4 haploid daughter cell. In mitosis, there's no crossing over is taking place. In meiosis, the crossing over is taking place. The recombination or the crossing over is occurring at the packaging stage of prophase 1 of meiosis 1. Mitosis generally occur in somatic cell, and meiosis occurs in gametic cell. In mitosis, there's no pairing of homologous chromosomes is observed, but in meiosis, the pairing of homologous chromosome occur, and the pairing of the homologous chromosome is known as synapses. Okay. And the significance of meiosis is that in meiosis, the crossing over takes place or the recombination occurs. And because of the crossing over, it leads to the variation. And the variation, they leads to evolution. Cell division or the cytokinesis in case of plant cell, it occurs by the cell plate formation. And in case of animal cell, it occurs by the cleavage furrow. Cell plate formation occurs from the center and then start moving towards the periphery. In cleavage furrow, it starts from the periphery and it moves towards the center. So that's all for meiosis and mitosis. I hope it is clear. Thank you all for your participation. Thank you all and see you in the next class.